So in this video, we're going to be talking about glycolysis. So glycolysis is a series of enzymatic reactions that takes place inside the cytoplasm. And in this series of enzymatic reactions, glucose, which is a 6-carbon structure, is going to be converted into two 3-carbon pyruvates. And in this process, we're going to produce ATP and NADH. ATP is going to be used as energy for the cell, of course, and NADH is going to be used as an electron donor in the electron transport chain to produce ATP. So these are the enzymatic reactions that takes place during glycolysis. Like we said before, we're going to start with glucose, which is a 6-carbon structure, and end with pyruvate, which is a 3-carbon structure. And these are the enzymatic reactions that happen inside of it. Now there are 3 that we're going to focus on in this video, and these are going to be the 3 most important. And that's going to be the conversion of glucose to glucose 6-phosphate, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, and the conversion of phosphoenol pyruvate to pyruvate. Now we can divide the reactions of glycolysis into 3 stages. The first stage is called the priming stage, and in this stage the cell invests energy in the cycle. The second stage is called the splitting stage, and here we're going to split the 6-carbon fructose 1,6-bisphosphate into two 3-carbon structures of glyceraldehyde 3-phosphate. The third stage is called the energy stage, and during these reactions we're going to produce ATP. So the first important reaction to know is going to be the conversion of glucose to glucose 6-phosphate. Now this reaction could be catalyzed by 2 enzymes, either hexokinase or glucokinase, and this depends on the tissue that you're in. So glucokinase is going to be found in the liver and in the beta cells of the pancreas, and hexokinase is going to be found in pretty much any other body tissue. Okay, so now we're going to discuss the hexokinase and glucokinase in more detail. And we're going to start with hexokinase. So hexokinase, as like we said before, is going to catalyze the reaction that converts glucose to glucose 6-phosphate. Okay, hexokinase is going to be negatively inhibited by rising levels of glucose 6-phosphate. So the higher the glucose 6-phosphate, the more inhibition that we have on hexokinase. This is important because it's going to prevent cells from taking up too much glucose and depriving other tissues from glucose. Now, it's important to know that when glucose enters the cell and is converted to glucose 6-phosphate, this glucose 6-phosphate cannot exit the cell. So it's important for cells to take in only the glucose that they need so they don't deprive other tissues from glucose. And insulin has no effect on hexokinase. Okay, so like we said before, glucokinase is going to be found in the liver and in the beta cells of the pancreas. Now, glucokinase is going to catalyze the reaction that converts glucose to glucose 6-phosphate, but it's not going to be inhibited by glucose 6-phosphate. It's actually going to be inhibited by the next byproduct of glycolysis, or the next intermediate of glycolysis, which is going to be fructose 6-phosphate. So fructose 6-phosphate is going to have negative inhibition on glucokinase. Now this negative inhibition can be overcome by rising levels of insulin and glucose. So how does this happen? So let's say a person after a meal, he's going to have high levels of glucose in his blood. And of course, high levels of glucose is going to lead to high levels of insulin. Now remember that glucokinase is normally going to be inhibited by fructose 6-phosphate. But when there's high insulin and high glucose levels in the blood, this is going to overcome the inhibition by fructose 6-phosphate, and it's actually going to stimulate glucokinase. Now this is important because after a meal, when you have all that extra glucose in the blood, you'd want the liver to take that glucose in and convert it to glycogen and store it. Now, let's say a person is in a fasting state, so he has very scarce amounts of glucose in his blood. So low glucose in the blood, that means there's going to be low levels of insulin. So in this case, fructose 6-phosphate is going to be free to inhibit glucokinase. So in this situation, the liver is not going to be taking up any glucose from the blood. And this is important because you want that scarce amount of glucose to remain in the blood and supply vital organs like the brain. Okay, so how does fructose 6-phosphate actually regulate glucokinase? Well, we said before that in the presence of fructose 6-phosphate, glucokinase will be inactive. So how does this happen? Well, in the presence of high levels of fructose 6-phosphate, it's going to stimulate a protein called GKRB, or glucokinase regulatory protein. Now this GKRB protein is going to bind to glucokinase in the cytosol, and then it's going to transport it all the way into the nucleus. And of course, this is going to make glucokinase inactive. We also said before that in the presence of high levels of glucose, the inhibition by the high levels of glucose could overcome the inhibition of fructose 6-phosphate to glucokinase. And how does this happen? Well, glucose is going to bind to GKRB protein, and it's going to prevent it from binding to glucokinase. So glucokinase is going to remain in the cytoplasm, and it's going to be active. Okay, so now we're going to talk about hexokinase and glucokinase in the setting of high blood sugar. So when there's high blood sugar, so this happens after a meal, the cells of the body are going to take in as much glucose as they need, and they're going to convert it to glucose 6-phosphate. Now, the increase in glucose 6-phosphate is going to inhibit hexokinase. So after a meal, hexokinase is pretty much going to be at low activity levels. Okay, so we said before, when there's high levels of glucose, and that's going to lead to high levels of insulin, this is going to stimulate glucokinase. And this is going to prevent the inhibition of glucokinase by fructose 6-phosphate. So after a meal, the glucokinase in the liver is going to be very active. And this is going to be important because you want the liver to take up all that excess glucose and store it as glycogen. Now, hexokinase and glucokinase in the setting of low blood sugar is the opposite. So when there's low blood sugar, that means there's going to be low glucose entering the cell, and there's going to be low glucose 6-phosphate. So hexokinase is going to be very active, meaning that the cells of the body are going to take in as much glucose as they can. On the other hand, glucokinase is going to be inhibited by fructose 6-phosphate because we don't have any glucose or insulin in the blood to activate glucokinase. And this is going to prevent the liver from taking up the scarce amount of glucose that is found in the blood. And this is going to spare the glucose for other organs of the more vital organs of the body, like the brain, for example. So now we're going to discuss the enzyme kinetics of hexokinase and glucokinase, and we're going to begin with hexokinase. So we're going to plot a graph, and we're going to put the substrate concentration S against the velocity. Now the substrate concentration in this case is going to be the concentration of glucose in the blood, and the velocity refers to the maximum rate at which an enzyme can produce a product. So in this case, it's going to be the maximum rate at which hexokinase can turn glucose into glucose 6-phosphate. So we're going to notice that basically hexokinase is going to have a low Km and a low Vm. Now, Km refers to the concentration of glucose in order to reach 50% of the Vm. So as we can see, a low Km means that it doesn't really take that much glucose concentration in order to reach 50% of the maximum velocity. So in conclusion, any glucose concentration, at any glucose concentration, hexokinase is going to reach maximum velocity. Now, to correlate this with glucokinase, so glucokinase is going to have a high Km and high Vm. So high Km meaning that we're going to need a high glucose concentration in order to reach 50% of the Vmax. And this indicates that there's going to be a large variation in glucokinase activity depending on the glucose concentration. Now, the maximum velocity that glucokinase can reach is very high, meaning that the maximum rate at which glucokinase can convert glucose to glucose 6-phosphate is very high. The last thing that you have to notice about this graph is that it's a sigmoidal curve. And this means that glucokinase has a cooperative characteristic, meaning that when the first glucose molecule binds to glucokinase, it's going to be easier for the next glucose molecule to bind and produce product. So an important clinical correlate to the first step in glycolysis is glucokinase deficiency. Now we said that glucokinase is going to be active in the liver and in the beta cells of the pancreas, and it's going to be responsible for converting glucose to glucose 6-phosphate. Glucokinase is going to be activated when there's high levels of glucose in the blood, so after a meal, and also when there's high insulin levels. Now in these patients, they're going to have a deficiency in glucokinase, meaning that their liver cannot take up as much glucose as it used to. And of course, this is going to lead to symptoms of hyperglycemia. In addition, glucokinase is also found in the beta cells of the pancreas. And glucokinase is very important in order to stimulate the beta cells of the pancreas to release insulin. So if you have a deficiency in glucokinase, you're going to have a deficiency or a decrease in insulin release from the beta cells of the pancreas, and this is going to exaggerate the hyperglycemia. And this usually happens in patients after meals.